In this video, I'm going to talk about dendrograms and cluster analysis. Dendrograms are a general group of trees. They aren't necessarily phylogenetic trees. And cluster analysis are a way that dendrograms are made. So there's many different ways that phylogenetic trees can be displayed, many different types of information that can be placed on phylogenetic trees. Dendrograms are tree-like, or rather phylogenetic tree-like diagrams that represent groupings or similarities between groups. They are not phylogenetic trees. Anything can be put onto a dendrogram. And scientists have been making dendrograms for a long time, often without any formal taxonomic system, or definitely without any evolutionary systems. For example, this tree here is from Lamarck, so it predates the insights of natural selection, evolutionary descent that Darwin had. So dendrograms are a general class, a general group of tree diagrams that are used throughout sciences. And you can put anything on a dendrogram, and you can group or classify anything. So key thing with dendrograms is there's no hierarchical structure, there's no nested structure. They're just for organizing information about similarities and differences. So we could group amino acids onto a tree if we wanted to. Now, amino acids, there's 21 of them. There's no hierarchy, there's no nested structure, there's no natural organization. They're just macromolecules that have similarities and differences, just like many objects in nature. So we can look at them and compare and contrast them and see how they might be grouped together. For example, we can look at which ones are positively charged, which ones are negatively charged, which ones have no charge, those that are hydrophobic, those that are hydrophobic and have rings. There's no hierarchical structure to this. These are just groupings in general characteristics. We could use kind of like a circle diagram or Venn diagram to show how all these amino acids are related. So all these different circles represent relevant groupings, and it's very complex, and there's lots of overlap because each amino acid can fall into different groups, and there's no hierarchy, no structure. So over here we have isoleucine, leucine, and valine are over here, all small hydrophobic molecules. Phenylalanine, tyrosine, and tryptophan are over here. These are large hydrophobic molecules. These are both these groups here, valine, isoleucine, and leucine, phenylalanine, tyrosine, and tryptophan. They're next to each other on this diagram. All of this space is occupied by different amino acids. So the hydrophobic amino acids are close to each other, but there's separation here, probably because of the size difference between them. So they're all hydrophobic. They're on the same end of the diagram. They're somewhat separated based on size. Now over here we have histidine. Histidine is not hydrophobic. It groups up with phenylalanine and tyrosine based largely on size and the fact that it has a ring, though it has a five-sided ring structure rather than a six-sided ring structure. So overall, histidine has some chemical similarities to these other ones over here. Now, if we want to, we could put this into a tree diagram. This does not imply that there's any sort of hierarchy or any nested structure. It's just putting things into a phylogenetic tree-like system where things that are chemically similar are grouped together and share a node. Things that are more distantly related, or not related, but that share fewer chemical similarities, share a different node. So isoleucine, leucine, and valine are kind of like in a clade over here. Tyrosine, tryptophan, and phenylalanine are a clade here. All of them are hydrophobic molecules, so they are in a larger clade, and then histidine is much more distantly associated with them. Dendrograms are built using statistical algorithms called cluster analysis. We won't worry about how that is actually done. This cluster analysis does not imply or does not think about in terms of hierarchy or nestedness. It's just all about grouping things and attaching them to nodes based on overall similarity. Cluster analysis, you will run into it a lot. You will see diagrams in lots of scientific papers: bioinformatics, genomics, proteomics, transcriptomics, metabolomics, in general data science, computational biology, statistics, some branches of ecology. All of them use cluster diagrams. And when you see one of these diagrams, you need to recognize that it's not a phylogenetic tree, that it is just based on relationships. So it is using some of the logic of trees and connecting similar things by node, but it's not indicating anything about descent or actual relationship or nestedness.